Thanks for coming. We've got some guests here too. Um, so Logan, if you went to Logan's proposal defense, you'll probably notice it's a little bit different. And one of those reasons is because we took a job with Kentucky State Nature Preserve's Mike Compton, who's here. He's an alumni, EKU alumni. So we ended up switching gears. So his thesis is a little bit different, but it worked out. This was a project we had sitting on the shelf. It was funded by Missouri Department of Conservation. Steve McMurray is here with us, who is the money bags behind this project, I guess. Many years ago, we had some contamination nightmares, and so the project got delayed, and Logan decided to pick this up since it was sort of a project on the shelf. Logan's from Somerset, and he is a graduate of EKU. He worked with Stephen Richter for a few years on wetland projects. As I said, he took a tech position with Kentucky State Nature Preserves for a few years. And now he's got a position at Kentucky Division of Water, and he's finally knocking out the defense here. So we're going to hear about his work on Ozark pigtoes.
You want me to share my screen now?
Yeah.
Okay.
Should now be able to share, Logan. Yeah, we're good. We see.
There we go. See everything going on here?
Yes.
Okay. Let me minimize. There we go. All right. Thanks everybody for coming. It looks like we got a pretty good crowd here. So I'm Logan Phelps, and today I'm presenting my thesis work on genetics and morphology in the genera Fusconaia and Pleurobema from Ozarks drainages. So a little bit of background on the area. This is an interior mountainous region within mostly Missouri and Arkansas. You can see it dips a little bit into Oklahoma and just a small hair into Kansas. But the mountainous topography of this region isn't quite associated with the tectonic uplift that we normally think of with mountains, but instead is the result of erosional processes acting on what once was a broad high plateau. So it's hypothesized that this area, along with the Ouachitas to the south and the Eastern Highlands that many of us here in Kentucky are more familiar with, used to be part of a more contiguous pre-Pleistocene highlands area that was basically split due to the formation of the broad Mississippi and Arkansas river valleys. So this area, you can see looking at the map here, drains radially into a handful of major systems, and it's entirely within the Mississippi River Basin. To the north, you have the Missouri River system with some major tributaries, and to the northeast, the Merrimack system. To the southeast, the St. Francis, and to the south, it largely drains into the White River system. And then just to the west, it becomes drains into the Middle Arkansas River system. And streams in this region are typically characterized as kind of cool, clear water, high gradient systems. So Maiden in the 80s published quite a bit on fishes of the interior highlands and published on that common geological history that I touched on. And so for this figure from Maiden 1988 kind of shows, gives you a picture of some of the high rates of aquatic endemism that we see associated with all three of these areas. And then that common geological history has also created some biological similarities across these regions. And there are some, I guess, themes as far as these faunal patterns go. And so on the top here, you can see a cladistic analysis that Maiden published in the 80s, looking at fish distributions and faunal similarities. And you can see that this interior highlands clade that includes Ozark streams as well as Ouachita streams, and they sit really similarly to a lot of what was associated with the old Taze River. Nelson in 2008 published similar cladistic analyses splitting Plecoptera fauna by state and province within North America. And you can see as well here, Missouri, Arkansas, and even Oklahoma were very similar in fauna to, again, some of those, the same region where we have Kentucky, Illinois, Indiana. And then Haag in 2010 analyzed mussel faunas split by major drainages. And here we see a little bit of an interesting pattern as far as mussel faunas from Ozark drainages go, where southern Ozark drainages like the St. Francis and the White System were much more similar to Mississippi embayment and Western Gulf streams, whereas northern drainages such as the Osage, Merrimack came out closer to some of these upper Mississippi streams as well as the Ohioan streams. And so we're really from here going to dive a little deeper into the mussel fauna and provide a little more context on that. So when we're talking about freshwater mussels today, we're talking about the group, the Unionoida, and that way we're excluding the groups that include fingernail clams and our species that are invasive in the US or in North America, such as Asian clams and zebra mussels. And so the Unionoida is a nearly worldwide distributed and a pretty diverse group, but really the center of that global diversity is in the Southeast United States. You can see from the numbers there on this slide that species in North America, this is per Williams et al. 2017, which was the last major taxonomic synthesis, that species are very unevenly split between the Unionidae and the Margaritiferidae. The most current literature, I think, puts estimates at about 10% of species within this group already having gone extinct, and close to half warranting protection, status elevation, at least to, to recognition as a threatened or vulnerable species. So within the Ozarks, there are more than 50 species already known. And just like any group, kind of the basis of effective conservation is a working knowledge of what species are and correctly delimiting species as best that we can. And so a lot of the taxonomy in Unionids dates back to the 1800s, and many of our contemporary names are still associated with naturalists of the time like Isaac Lee and Rafinesque, just to name a couple. And a lot of the historical descriptions base information largely on shell material. And the reason for that is shell material is readily available and easy to find for especially thick-shelled species. Once the animal has expired and that soft flesh rots away, these shells can persist on banks and bars and just in the environment for years. We now understand better than we knew in the 1800s that shells are very variable based on environmental factors. Ortman in the early 1900s published on variation based on longitudinal stream position. And so that is just as you go from a lower order stream to a higher order stream. And we know that substrate type, which tends to be associated with stream position in most cases, as well as water chemical parameters, can affect these shells' developments. And so because of this natural morphological variation, there have been a lot of problems within Unionid taxonomy. A lot of these problems go back to a couple of major themes relating to cryptic diversity, where you would have multiple lineages that are so morphologically similar that we have described them as the same species. And then cases of morphological variation where the variation within one lineage is so high that it's been described as multiple species. A lot of these issues went undetected until we started using more molecular analyses on the group. With that variation in morphology, a lot of researchers in malacology have moved to the use of geometric morphometrics to analyze shell shape. So traditional morphometrics would rely on measurements and ratios, whereas somebody might take the length over the width of a shell and use ratios like that to compare shell shape. Geometric morphometrics relies more on landmarks and the measurements and angles between them. There are different types of landmarks, and in some groups, such as cypriniform fishes, you can see this figure on the bottom left. What we would call a type 1 landmark can be standardized, and this is type 1 just refers to a true homologous structure. For example, number 11 there, kind of the anterior origin of the dorsal fin. If you're looking at any cypriniform fish, you can plot a point on that dorsal fin like that. However, mussel shells largely lack those types of landmarks on the exterior, especially. On the interior, you might, I think some researchers have landmarked on the hinge teeth and mussel scars and those types of features. But on the exterior of the shell, there's really not a lot to work with in that way. And so we get into the concept of type 2 landmarks to describe shell shape. So type 2 landmarks on shells are typically plotted based on equal intervals or equally spaced rays bisecting the shell's edge. Sometimes type 2 landmarks are curvature maxima and minima. And this technique has been applied to bivalve shells in other research, as well as gastropods. This technique in the literature has been shown to be able to differentiate kind of minute differences among very similar shells. And so now you might be wondering what this has to do exactly with Fusconaia pleurobema. It's a little bit of broad context, and we're going to narrow down in to this group, the focus of my study. And so here's just a slide for you showing multiple species and some of the different shell shapes that we see in these genera. So when we talk about pigtoe mussels, we're talking about the Pleurobemini, which is a tribe that is a pretty significant chunk, about a third to a fourth, or a fourth to a third of North American freshwater diversity. In Williams et al. 2017, that last taxonomic synthesis, you can see that a lot of the diversity in this group was really put into or accepted into 3 genera, Elliptio, and then our focus today, Fusconaia and Pleurobema. A lot of these shells lack very hard, distinct features, such as pustules and nodules that you would see more in like the quadrillini. And a lot of species even lack colored rays that can be taxonomically informative. There's obviously exceptions to that, but many of them, you might be tempted to describe them as very plain shells. They've kind of developed a reputation for being difficult to reliably identify. If they don't have that reputation with everybody, they definitely have it with me. And it just relies mostly on the shape of the shell because of that lack of features. So there have been a couple of studies that have given quizzes to professionals as well as workshop participants and found that even professionals just differentiating between Fusconaia flava and Pleurobema syntaxia, which aren't even congeners, we're not totally accurate. In general, the more experience a person has, they were better at it, which makes sense. But those studies didn't report 100% correct IDs. And so this leads us to some of the taxonomic problems within the Pleurobemini. And I'm going to talk about a couple of cases here that we're going to focus in on. And the first of those is that recent work has revealed cryptic diversity in the Ozarks, in mussels in general. There's a few cases of species that appear to be cryptically diverse in this area, but this also includes Fusconaia, which we're going to focus on. And then we're going to talk about a case where genetic data in Pleurobema have raised certain questions about the species delimitation there, and especially in regards to morphological variation. On the topic of Fusconaia, Burdick and White in 2007 published a phylogeographic paper where they looked at the cytochrome oxidase 1 mitochondrial gene, or CO1, across the species range, and it even included Fusconaia serina from the Mobile Basin. And so what they found were two unique haplotypes that they reported as F8 and F9. And you can see on the map here, they were centered in F9 came from the northern Ozarks, while F8 was over in the upper Arkansas Great Plains region. These shells, or these specimens, I should say, were collected as and would have been considered in the past as representatives of Fusconaia flava, but based on the phylogenetics here, appears to be a case of cryptic diversity. Now, Inouye et al. In 2018 identified the same lineage from the Illinois River in Arkansas. And so I've approximated that on the map with that big red star. So this also is an Arkansas River tributary. Campbell and Lydiard published in 2012 a paper that corroborated Burdick and White's findings and suggested that this lineage was appropriate to describe and name differently. And they recommended resurrecting the name Fusconaia hebetata as a name for this lineage. Now, this is currently considered a synonym of Fusconaia flava, and unfortunately, the existing type specimen has a very broad locality of Missouri. And so that could be North Missouri, South Missouri, in the Ozarks, out of the Ozarks. It's kind of, you just don't know from that. Inouye et al. In 2018 suggested possibly elevating the subspecies Fusconaia flava sampsoniana to the species level to name the species. And this was based on conchological similarities as well as a more precise type locality of the Elk River in Missouri, which is part of the Arkansas system, and so should theoretically include this F8, F9 lineage. So being that we don't know exactly what name is most appropriate, but one of these is probably applicable, I'm going to refer to this lineage for the rest of the presentation here as hebetata sampsoniana, because I haven't made a decision, I've been calling it both. And so to move into Pleurobema, we have a case here involving two species, Pleurobema rubrum and Pleurobema syntaxia. And so the problem with these species actually originates outside of the Ozarks, over their broader range in the Mississippi River Basin. You can see, looking at these lectotype photos on the right, that these two shells have pretty dramatically different shapes. Pleurobema syntaxia on the right there is the round pigtoe, and on the bottom, rubrum is the pyramid pigtoe. And looking at rubrum, I feel that you can really see why it gets that name, just with that tall, very triangular shape. And so these two species both have very different conservation needs. Pleurobema rubrum is very rare and is actually considered or petitioned for listing under the Endangered Species Act and the federal protections that come with that. Pleurobema syntaxia is considered much more stable throughout its range. However, genetic work just within the last decade has created a little bit of confusion about the species. Jones et al. 2015 looked at the ND1 mitochondrial gene and found that they weren't differentiated there, but most researchers tend to put more weight on the CO1 gene as the universal species level barcode. And so Inouye et al. In 2018 built on that work and found consistent results that Pleurobema rubrum and syntaxia were indistinguishable. And so that raises the question of whether this is a case of phenotypic plasticity or just more, some kind of morphological variation producing two morphotypes. Geographic sampling of this relationship has been fairly extensive and was covered a lot in Inouye et al. 2018, but some areas, including the Ozark Highlands, remains kind of a hole in the sampling area. And so to bring all that together, we wanted to focus on these relationships in the Pleurobemini and investigate them through the Ozarks. To be specific, the points of this study were to use molecular techniques to investigate the extent of Hebetata sampsoniana, as well as to investigate the relationship between rubrum and syntaxia in an area that has not been so well assessed. With genetically vouchered specimens, we also wanted to use geometric morphometric techniques to morphologically compare shell shapes and investigate how variable these lineages and species are. So we attempted through morphological data to also place the type specimens of Hebetata and flava sampsoniana that I showed you earlier, but this has been removed due to just some issues in our methods relating to I guess the photography of those shells that we haven't been able to get around yet. So our study area is the Ozark Highlands. All samples were taken in southern Missouri, and we had about 10 localities where mussels were collected by the Missouri Department of Conservation in Arkansas State University from 2012 to 2014. And these were all deposited at the ASU Museum of Zoology. We didn't get genetic information from all localities. However, we did touch on most of the major drainages. So tissue was preserved in 95% ethanol, and it was extracted using the CTAB protocol. DNA yields were standardized to 10 micrograms per milliliter. And then we used the polymerase chain reaction to amplify this CO1 mitochondrial gene for our specimens. Unfortunately, even though samples had been preserved at 95% ethanol, they had continued to degrade over time. And so we ended up having to amplify two sections of this region, basically the 5' and the 3' end, with a sequence gap in the middle, which is not ideal, but was consistent with some of the work that Burdick and White had published in that 2007 Fusconaia flava paper. So we were able to make use of the same primers that they used, as well as some Fusconaia ozarkensis specific primers. PCR product was visualized via gel electrophoresis to confirm that we got amplification, and then raw DNA was sent to Eurofins Genomics, where it was sequenced via Sanger reactions. Sequences were, or data that came back was assessed and trimmed in DNA Dynamo, and then sequences were aligned within AliView. We aligned our new data to the same data set that was published in the large phylogeny of Inouye et al. 2018, including the same outgroups, and we added some additional Fusconaia mitchellii sequences that were published just last year by Smith et al. We let model selection go in IQ tree and selected a model that IQ tree suggested was the most appropriate based on BIC. And then we used, again, the same IQ tree program to build a maximum likelihood phylogenetic tree. And this was utilizing the 1,000 ultra fast bootstrap method within that tool. In order to go, or in order to assess geometric morphometrics in the group, we photographed the right valve of all of our genetically vouchered shells. And that was done using a photo box so that shell distance to the camera lens could be standardized. And then the program's MakeFan8, or the program MakeFan8 was used to overlay an equally spaced fan aligned to 2 homologous points on each shell, and these in this case are right here the posterior end of the hinge ligament, as well as the vertex of the angle formed by the umbo and the anterior wing of the shell. And then TPS Dig 2 was used to digitize 2 type 1 homologous structures, which are the 2 I just said that we align the fan to, as well as 18 semi-landmarks where the fans crossed the edge of the shell. In order to capture inflation of the shells, the right valve, the same valve that we photographed, was placed on a radial grid and aligned to the 180 degree mark using the same 2 points. And then a contour gauge was pressed down onto the shell at 210 degrees in order to capture a good profile of what that shell would look like, almost more dorsally or ventrally. Again, the programs MakeFan8 and TPS Dig 2 were used. We overlaid a 30-ray fan, again 12 degrees each, and then digitized 2 homologous structures or landmarks and 14 semi-landmarks. The homologous points in this case are just the ends of the shell. A handful of specimens had to be removed, unfortunately, because they just had very excessively worn shells. But we also included specimens from the lot that Burdick and White sequenced their F8 and F9 haplotypes from as representatives of Hebetata sampsoniana. Landmark data was analyzed using the paleontological statistics software, in which landmarks were aligned using procrusty superimposition, and all that is a method of removing variation related to size and orientation, leaving you with only variation related to shape. Within PAST, we performed principal components analysis and linear discriminant analysis on the Procrustes coordinates. And linear discriminant analysis is just another ordination technique that looks for maximum separation of groups using an a priori grouping factor, in this case, the genetic identification of the specimens. LDA was also used to reclassify data points. So for the shell outlines, the PCA had 2 axes that accounted for just about 9% or 90% of the variation in the data set, and all other axes were less than 10% individually. We saw a lot of variability in or variation in the Hebetata sampsoniana group, as well as the rubrum and syntaxia group. There was high overlap between all groups, and with Ozarkensis and flava being the only 2 that were distinct from each other. They had absolutely no overlap. And so here's our PCA plot. You can see this listed here as Fusconaia spa is that Hebetata sampsoniana lineage. And there's quite a bit of overlap there with rubrum and syntaxia. We have Fusconaia ozarkensis kind of up here by itself. Sorry, that was my cat And here we have Fusconaia flava that is a lot more compact and by itself. The linear discriminant analysis yielded 3 canonical variates that described, the first one described just almost 60%, and the second variate described just over 30%, and then the last at just over 10%. And in this plot, Fusconaia flava was easily removed from the other groups, and there was most of the similarity in the plot was between Hebetata sampsoniana and Fusconaia ozarkensis. The model here reclassified points with 100% accuracy. And one thing you want to notice from this table here is that we had very few representatives of, I should be doing this way, of ozarkensis and flava. And that you can see that in the plots how there's less points making up those clusters. So here is the plot of that LDA analysis. You can see a lot of overlap between ozarkensis and this Fusconaia species, as well as, again, a large, highly variable rubrum syntaxia group. And Fusconaia flava here very distinct by itself. The conclusions that we can draw from this is that the PCA overlap would suggest or reinforce the idea that these are morphologically similar shells that and thus identifying them can be difficult. Although the LDA reclassification was 100% correct, I do have some hesitations about putting too much emphasis on that, which I'll get into once I finish the rest of the morphological data. You can see most of the variation that we saw in groups was related to rubrum syntaxia and Hebetata sampsoniana, which again, I have some similar reservations about that I'll get into. But we were able to look at what the changes in shell shape were like along these PC axes, whereas PC axis 1 was related to shells with an elongated and narrowed posterior margin, and also having a more broadly rounded anterior margin, shifting to a more bluntly squared shape. You'll see on the thin plate splines that there's also a shifting along the ventral margin of the shell, kind of related to concavity, while PC2 tended to be associated with a shift from an overall triangular shaped shell to one that is much more ovoid shape. So here's our thin spline deformation grids, as well as a couple of representatives of what we're seeing going on here. So you can see from these two shells, negative on PC1 axis 1 is this left specimen, and positive is on the right. You can see how the shell kind of compresses in the anterior posterior direction, becomes just kind of more square to triangular. Here is the same thing looking at specimens from PC2. You can see this much more, the specimen on the left is kind of rounded overall. It was a small specimen, but has the beginnings of that triangular shape, going to what we see on the right, which to me looks more like what I think of as kind of a typical Ozarkensis, I guess, if you can say that. Looking at shell inflation, we again saw Ozarkensis separated from all other groups in the PCA plot, and that theme of Hebetata sampsoniana and rubrum syntaxia being just highly variable is prevalent here again. PC axis 1 here accounted for over 80% of the variation, axis 2 accounted for almost 10%, and all other axes were less than 4% of variation individually. So here's our PCA plot. This is again based on the shell inflation curves, and you can see very similar to before, a high amount of overlap between the Fusconaia, Hebetata sampsoniana, as well as the Pleurobema rubrum syntaxia group. Ozarkensis and flava are both, Ozarkensis is very much distinct by itself. Flava has a little bit of overlap there with both of the other groups. In the LDA model, we had 3 distinct canonical variants explaining 57, 37, and about 5.5% of variance. And reclassifications in this model were less accurate than in the other model. There was more confusion here entirely between the Hebetata sampsoniana group and the rubrum syntaxia group both ways. And so this confusion was reflected in the ordination plot here, where flava and Ozarkensis are very much distinct off by themselves. And then you have just a lot of overlap between rubrum syntaxia and Hebetata sampsoniana. And so the overlap in the PCA plot to me suggests high morphological similarity among these shells. And the LDA reclassification was a little better at, I guess, showing the same thing here with the confusion of those Hebetata sampsoniana and rubrum syntaxia specimens. Here, PC1 tended to be associated with a shift from very compressed and evenly rounded shells to those that were inflated and even a little irregular. Maybe they had a shallow sulcus that produced kind of a little sinuosity in the profile. And PC2 tended to be associated with a shift from flat, steadily sloping anteriors to those with a more evenly rounded profile. So here are the thin spline deformation and examples showing as PC1 changed from negative on the left towards positive on the right. And you can see definitely the inflation there, as well as that bit of irregularity here near the top of the shell where that contour gauge pressed into just a little bit of a shallow sulcus. Here is the same thing for PC2, where you can see a very steadily sloping shape compared to a much more convex, evenly rounded shape. And so a little bit of that caution that I mentioned earlier, the results of these morphological analyses, I think should be taken with a little bit of caution, as you noticed sample sizes in this analysis were very uneven. We had small sample sizes in Fusconaia flava and Ozarkensis, and as well as limited collection localities. And so that in itself, I think would bias flava and Ozarkensis towards some of the distinctness and less variation that we saw. If we had representatives of those from different size streams and different systems, we would see, I think, a lot more variation and a lot more overlap in our ordinations involving those species. The LDA model, it's interesting to note, just when it came to shell inflation, suggested confusion between this Fusconaia lineage and the rubrum syntaxia group, although members of this Hebetata sampsoniana lineage have been identified as flava or Ozarkensis, or even sometimes just left at the genus level.
You're at about 10 minutes. You got about 10 minutes.
Oh, okay. So here is some of our genetics results. I'm omitting the Pleurobema tree here, as it's just a massive mess that can be really summarized with one clear sentence. So what we see here, or what we saw in our tree, was specimens of flava and Ozarkensis were only collected from the White River system in southern Ozarks. Everything that we collected that was a member of Fusconaia from outside of the White River system clustered with this Hebetata sampsoniana clade, and in that you can see Burdick and White's F8 and F9 specimens. Pleurobema rubrum and syntaxia were found in all drainages and showed absolutely no differentiation at this CO1 region. So the conclusions we can draw from that regarding Pleurobema was that we confirmed the presence of the rubrum syntaxia clade in all major drainages, which is not surprising. We knew that, or I guess suspected that they were there. Rubrum and syntaxia in the Ozark Highlands are genetically indistinguishable from each other, which is consistent with the issue that we're seeing over their entire range. And all this study did was help kind of fill in a knowledge gap regarding that relationship. For Fusconaia, we now have evidence of Hepatota sampsoniana in every major river system of the Ozarks, except for in the south, in the White River system. It appears to be flava and Ozarkensis. So this is consistent with Burdick and White, and in a way, and builds on their results to slightly expand the geographic extent of that lineage. We also wanted to touch on the possibility that flava and Ozarkensis are potentially absent totally from the region, except in the White River system. It's very peculiar that representatives of those two species were only found in one system. In light of that, we think it may be appropriate in the future to reevaluate surveys in the area, as well as life history studies. For example, in 1997, there were descriptions of conglutinants and fish hosts for Fusconaia ozarkensis out of the Arkansas River system. It's possible that those could have been descriptions attributable to Hepatota and sampsoniana. We can't make any fine scale biogeographical inferences, mostly I think because of our large sequence gaps. There was no structuring within the phylogeny. We do have at least 3 possibly isolated units within the Arkansas, Merrimack, and Missouri River systems. The only contemporary connection between those would be the main stem Mississippi. And this distribution of the North and the West Ozarks is something that has been observed in other species. For example, Ethiostoma microperca has a similar distribution. We wanted to touch on a strange relationship that we noted here, a more broad relationship where this clade's closest relatives are Western Gulf Texan species related to like the Neches and Sabine, Trinity rivers. And so we were speculating that this might suggest a prehistorical connection between these drainages. And kind of found a little bit of information on that in the form of Metcalf's 1966 hypothesized ancestral plain stream. This would have been a Pleistocene stream or pre-Pleistocene that drained a lot of the Great Plains in a north-south fashion. So here's a figure that Metcalf published, and number 1 right here would be this ancestral plain stream that reaches up into the headwaters of the Missouri and Arkansas River system. Now, this has been used to explain distributions of things with disjunct populations like Fungulus and Orconectes species here, although some researchers have refuted its existence based on geological information. Here's just another figure showing Maiden's 1988 reconstruction that includes the same stream here, again reaching into the headwaters of both the Missouri and the Arkansas River system. And a little bit of ambiguity here regarding where West Ozark streams might have drained to, whether they were Mississippian or would have potentially went westward. Elfrink in 2007 suggested an alternative here in the form of a Taze River that interestingly goes, instead of meeting the Mississippi River or going down through the Wabash Valley, would have gone over the north and to the west of the Ozarks, eventually draining into the Sabine. And so this would provide a very direct explanation for the relationship of those clades. But this publication hasn't picked up a whole lot of reference and support in the literature. In the future, we would like to continue to sample rubrum and syntaxia over their broad ranges and investigate them using different genetic techniques, look for evidence of gene flow between them, as well as continue to investigate Fusconaia over the whole Ozarks area to completely understand the geographic boundaries of those genetic lineages. It would also be nice to obtain more vouchered specimens of flava and Ozarkensis for more robust and meaningful morphological comparisons, and to get fresh photographs of the Hebetata and Sampsoniana type specimens that I showed you earlier to be able to morphologically compare those. It sounds like I'm pretty close on time, so I won't bother you all with too many shell pictures here. This just shows a lot of the variation. And with that, I want to thank my advisor, Dr. David Hayes, as well as committee members, Dr. Braccia and Dr. Richter. EKU, MDC, and ASU provided multiple levels of support in this project. And John Harris, Scott Feynman, and Steve McMurray in particular provided support from the field to museum accessioning. So with that, I would take any questions. Should I go back unshare or should I keep shared?
You can unshare for sure.
Oh, let me pause share. Stop share. Okay. Cool.
Logan, I've got a question. So, first of all, great job, great presentation, wonderful analysis.
Thank you.
Could geography, and this is very dicey here, I realize, but I'm put it out there. Could geography be a consideration in identifying a particular species? So say you have a particular shell, and could be this, could be that. Would you feel comfortable, based upon your analysis, saying, well, based upon the geographic distribution or locality, I would put it in this group? Or do you feel that there might be some groups that ignore the geographic boundaries and might be found in different, that is, how tight do you think the geographic association is with your particular species?
I mean, to collect a shell, there, as far as unionids go, there are a lot of tricky specimens. And I think it happens a lot for shells to be collected and say, based on the drainage we're in, this should be this species, that's what we should call it. And the same thing I think happens with almost any study organism, where you have cases of morphological similarity. A lot of times I think that those are things that have been, talk about splitters, and so they've been split based on genetic evidence. And so that's kind of what I think, I was alluding to at the end there, that I think more molecular work is warranted just to better understand and see if there are places where these Fusconaia lineages are occurring together, or if it is exactly as cut and dry as that would seem to suggest.
Thank you.
Yeah. Anybody else?
Yeah, following up on Dr. Calley's question, based on all of the morphological data that you collected, is there a practical application of your findings for the field biologist who's out there picking up shells?
Well, definitely the morphological data goes to show that there's a lot of overlap in the shapes of these shells. I think there have been some efforts to create models that can take landmarks and outlines and accurately provide an identification to somebody that needs that identification, the field biologist. But in general, these results are kind of very descriptive, and I think for the field personnel would be something to look at and say, okay, we see a lot of overlap between these two species, and maybe these fine details of variation in this way on the posterior end, or more concavity along the margin there, higher umbos. I think that it can be used descriptively, but does reinforce that they're difficult by nature.
Okay, I have a follow-up question. Going back to Arnold Ortman's work on longitudinal variation, I know with Ozarkensis and flava, you don't have really enough specimens to look at this, but with others, did you happen to look statistically or whatever, if there was a variation, if there was variation as you went from upstream to downstream in terms of morphology?
It's kind of hard to do that with our data set, considering that sampling locations within each drainage were very limited. You know, we would have one area from, say, the Burbus that we had specimens from, and one area of the Osage River and et cetera. We haven't looked at combining that data set in that way, just based on, you know, I guess we could look at the drainage size in square kilometers or miles or something based on where those samples came from, but we haven't looked at it that way yet.
Okay, thanks, Logan. Nice job.
Yeah.
Logan, a question for Dr. Hayes, I guess, and also you. So David, do the mussels exhibit biparental inheritance of the mitochondrial genome? Is that correct?
Logan, go ahead.
Yeah, so it's been a minute since I talked about this, but I know that there are two mitotypes of that CO1 region, whereas if you have a male specimen through the whole body, as my understanding, would have the female inherited mitotype, whereas the male in you know, only in its reproductive parts would have the male inherited mitotype lineage. And so that is another thing, is that all of this work is based off of that maternally inherited mitochondrial lineage.
I'm sorry, but if it's biparental, I'm sorry, but don't you have both maternal and paternal? The question I'm getting at is, does the biparental inheritance really complicate, or is there something that really complicates a bit of the analysis you're doing if it's biparental inheritance versus uniparental inheritance, say maternal in most organisms?
Well, a lot of this CO1 mitochondrial is based off the maternally inherited mitochondrial genome. And so if you're taking tissue samples from, say, the foot, which, you know, is common in what we did here, it would all be the same maternally inherited lineage. Is that more what you're asking?
Yeah, you'd have to sample the gonad to get to the male mitochondrial lineage, which we have not sampled in this study.
Okay. Yeah. Yes. Thank you, Logan. It was more my ignorance of which tissues have which genome. But thank you.
Okay. Yeah. In my understanding, just in the literature, there's just not a lot published on that male inherited lineage. Dave can correct me if I'm wrong on that.
Hey, Logan, I have a question for you regarding the rubrum syntaxia situation. Going forward, what would your recommendations be for handling that and for management purposes?
Could you clear up what you mean by handling that?
Are you comfortable saying they are the same species and they should be treated that way, or should they be treated as still two distinct species?
I mean, it's a difficult question, I guess. You know, if you're considering what the Co-1 evidence is, it definitely seems like they are the same species. You know, if you're doing a survey and making collections and field notes, you know, it certainly couldn't hurt to keep track of you know, you would call one maybe quote unquote syntaxia or quote unquote rubrum, you know, for informational purposes. But functionally, I think that there is still a little bit of work to be done. Like I said, you could look for, or some researchers need to move into things like population genetics in order to see if there's gene flow between the two species, if they're reproducing together. I think that it's a little bit of a careful situation just because of the conservation implications associated with rubrum particularly.
Yeah.
I don't know that I would make a hard and fast recommendation on whether you need to consider them totally separate or the same, but I would say that based on the evidence we have so far, it seems like they're the same.
All right, thanks.
Yep.
Good job, by the way.
Thanks.
Logan, just one final comment on my part. I love how David's group combines these biogeographical questions with taxonomy, with systematics. I think it's really cool to look at distribution of species and tie it back to ancient geologic events like the Taze River. So that's fantastic. And so great job combining those two elements, because again, the taxonomy may give us some insights into ancient river drainages and things like that. So that really is cool, I think.
Thanks. Yeah, I got pretty excited whenever I made the connection that, you know, we might be able to draw a little bit of that in based on these results and that genetic relationship. Unfortunately, about the ancestral plane stream is it seems like it's much harder to dig up information on it than say like the Taze River. It's been published on a lot more, but there's stuff out there, and certainly it raised a lot of interesting questions to look at those pieces together.
Anything else for Logan?
I'll throw out there real quick that you know, our current wisdom that we follow is that Fusconaia in the White River system is Ozarkensis and Ozarkensis only to address Dr. Callie's question. And so it's interesting that you picked up specimens that were, you know, were turning out to be flava from there, because John Harris and I did find some oddball specimens that we couldn't really place, and often Ozarkensis is confused for headwater form flava, and so I guess I'd like to see, not that I disagree with your findings, but I'd like to see the specimens, you know, the shell outlines of the specimens that came out to be flava, because that's pretty interesting.
Mm-hmm.
You know, for our work here. So, and most of my work going forward, if it's something in the White River system, I call it Fusconaia Ozarkensis, and anywhere else in Missouri, it's just Fusconaia spa.
Yeah.
Until the Hebetata Samsoniana flava issue is resolved. Did you include the specimens that were the original Osage River at Tuscumbia Hebetata specimens? Were those in there?
That's from Burdick and White's publication.
Yeah.
We included those. I guess I could have, or maybe should have mentioned a little better that because they didn't in that publication allude to, you know, those are lots of multiple specimens. And so since we couldn't from their pub get exactly which ones they had got DNA from, we know based on their numbers that it wasn't the whole lots. But we did go ahead and operate on the assumption that those lots, being that they all came from right there together, were all representatives of the Hebetata Samsoniana. But we did include those in the morphology.
Logan, I just wanted to say hi.
Hey.
How's it going? It's good to see you wrapping up and everything. That's so exciting.
Thank you. I appreciate it.
I was going to mention to Steve there, Logan's operating on a very broad definition of the White River. So where do your flava samples come from? Do you remember?
You talking to me or Logan?
Oh, Logan.
Okay, all right.
Oh,
no, I don't remember.
I would honestly have to look it up off the, open up my files and look back into it. I don't remember off the top of my head, but they weren't.
They come from the Upper Black River.
Oh yeah, that's right. I do recall.
So you're talking, technically it's the White River drainage, but it's like way down there.
Yeah.
And, so Steve's probably not talking about Black River when he's talking about the White.
Yeah.
No, I'm talking about the core White, like James, Current River, 11 Point,
So Logan has not found flava in the White that you're talking about.
Okay, all right, perfect, good.
Right.
I was just speaking. Makes my life a lot easier, so.
Sorry about that confusion. Yeah, I was just speaking. I included it because the Black River is a large tributary of the White, though.
Yeah.
Since they're in the system, I figured for simplicity's sake, I would just define that whole unit as the White River system. So sorry about that.
Fair enough. No. Yeah. Nice job, Logan. Money well spent on our part. And before we get cut off, it's good to see all my EKU friends up here. Good to see you're all doing well. So
good to see you, Steve. Thanks for coming in.
Good seeing you, Steve.
Yeah, great seeing you too, Steve.
Yep.
All right, I think that wraps us up.
Thanks for inviting me, Dave.
Thanks for coming. Good.